Thank you, Muz, for sharing highlights with us of your work. We will indeed keep the questions for the end and move now to a presentation by Dr. Catherine Blish. Dr. Blish is an associate professor of medicine in the Division of Infectious Disease and Geographic Medicine at Stanford University, as well as an investigator of the Chang Zuckerberg BioHub. As a translational immunologist, the primary goal of Dr. Blish's lab is to develop new methods to prevent and control infectious disease through better understanding of human immunology. Today, she will share with us some insights gained into SARS-CoV-2 pathogenesis from studying host pathogen response at single-cell resolution. Dr. Blish, we look forward to your talk.
Wonderful. Thank you very much. I'm sorry, I was having trouble finding my my share. So I'm going to try and cover a fair bit of ground. So I'm going to go kind of quickly, and I'm not going to give a lot of background as to why we were interested in understanding SARS-CoV-2 pathogenesis, because I think we all know why we're here, and really just begin with the goal of what my lab was aiming to do with this. And our goal was to elucidate the immune pathways that on one hand might lead to protective immunity, and on the other hand, immunopathology in COVID-19. And really to do this, our long-term goal is to look at both mild cases, which might inform vaccine design, as well as the more severe cases, which might inform us as to what sorts of immunotherapeutics we might want to look into to interrupt this disease process. And really, we've been taking a two-pronged approach to this. And one is to perform in vitro studies of viral pathogenesis. And these have involved a variety of systems, but primarily including lung tissue and organoid infections. And I'll just give a very brief snapshot of some unpublished data there, as well as in vivo studies to assess immune function across this range of disease. So I'm going to briefly mention the SARS-CoV-2 infection. And in particular, in a collaboration between my lab and Calvin Kuo's lab, we've been looking at distal lung organoids. And this is work that's been primarily performed by Shannon Choi, a talented MD PhD student in his lab, as well as Arjun Rustagi, a fellow in my lab, who really began our SARS-CoV-2 program and got the virus growing very early on. And what Shannon and Calvin have been working on, along with many others in the lab, and this is up on the bioRxiv, is a system in which they can grow clonal organoids from human distal lung, and they can actually, depending on the culture conditions, differentiate these to be either relatively AT2 enriched or basal enriched or so forth. And of course, AT2 cells are the high ACE2 expressing cells that many think are the target of this infection in the distal lung. And in fact, what I'm showing here is a picture of such an organoid grown primarily of AT2 cells. And in the panels on the left, we can see a mock infection. And in the panels on the right is a SARS-CoV-2 infection, in which we've stained for SARS-CoV-2 nucleoprotein in the red. And you can see robust infection of that. And so that was nice and tells us that we've got a good system in which we can further explore pathogenesis, and we'll hope to have some nice single cell data coming down the road. What was a little more surprising was what happened when we infected these basal organoids, which have multiple cell types, including club cells and ciliated cells. And in fact, if you evert these so that they're growing naturally with the ciliated cells on the outside, what we observe is you can see nice differentiation. But in fact, most of the infection occurred within club cells, which were not previously thought of as a major target cell. So of course, we're working on this further. But one of the nice things is that this is really a medium throughput system in which we can use this to really test therapeutics and immune regulators in a way that might be more physiologic than just testing viro cells in a dish. So we're quite excited to develop this further. So I'm going to switch now and talk about what's happening in vivo. And this is work that's been primarily driven through a collaboration that I started very early on as the first cases came to Stanford with my pulmonary critical care colleague, Angela Rogers, as well as several of my other ID colleagues. And within my lab, Aaron Wilk, Arjun Rustagi, and Nancy Shao did most of the hard work to acquire this data. But importantly, my lab was actually the central clearinghouse for all these samples, and everyone in my lab, pictured here in pre-COVID times, was processing and helping with all these samples. And I'm going to skip through a lot of the details because this work was published, but just mention that what we did was we actually grabbed samples as we were processing them on the fly and ran single-cell RNA sequencing with the Seq-well platform. Now, it just so happened in this first snapshot of 7 patients with 8 samples, they were all male, which was a little disappointing, a wide range of ages. Somewhat more lucky, about half of them were in severe disease with acute respiratory distress syndrome, and half were not. And in fact, the one patient from whom we had longitudinal sampling, we had a sample both before and after he developed severe disease. So if we just look at the global snapshot, this is the single cell data from over 40,000 cells from these 7 patients and 6 healthy controls that were processed completely in parallel. I've colored the healthies in blue, the COVID samples in orange, and you can see the cell type distributions labeling on the clusters over on the right. And it doesn't really take much to notice there's a massive reconfiguration of the immune system. If we actually look through what cell proportions are changed, we observe quite a number of interesting things in the peripheral blood. The first was depletion of a lot of innate immune cell subsets. I'm highlighting here the frequency of monocytes, plasmacytoid dendritic cells, gamma delta T cells, and NK cells, more particularly in the acute disease, which we've colored with more intense colors here, or the most severe disease. We also observed, again, perhaps not surprisingly, a reduction in naive B cell proportion in the COVID patients with an induction of plasmablasts, because of course we don't expect to see these in healthy individuals. And we did see a very weird population of cells that happened to be proximal to the plasmablasts in UMAP space that was seen really in the ARDS patients, but expressed markers consistent with neutrophils. And here I'll mention again that one unusual thing we did was we actually ran these on freshly isolated PBMCs. So cells that would normally have died during cryopreservation ended up going in. So I don't have time in 10 minutes to go through everything, but I just want to mention a few highlights. Obviously, we were very interested in the monocytes because they are so changed. And we really observed, if we just targeted in on those monocytes, this reconfiguration. I'm showing just CD14 and CD16 standing down here, as well as IFI27, one of the interferon-stimulated genes. And you can see that the COVID patients, particularly those more severe, have a shift towards a more activated phenotype. Pardon me. In the panel on the right, I'm showing a heat map of the expression of the most upregulated and downregulated genes in the COVID patients compared to all the healthy controls. And we really observed that there are a lot of downregulated genes, mostly MHC class II genes, which was seen previously in both SARS and MERS and has been seen by a number of other investigators since. And upregulation of a number of genes, mostly, if you're familiar with this list, interferon-stimulated genes, but also some alarmins. And but what really surprises us and was perhaps preluded by some of Ben Tanover's and others' works is it's really heterogeneous. And if we'd only grabbed a couple of these patients, we would have perhaps concluded different results. What was somewhat more surprising was what we saw in terms of pro-inflammatory cytokine secretion, because we all know there's a lot of inflammatory cytokines in the setting of severe COVID, and even our more mild patients were quite severe. But we really saw very little production of cytokines at the transcriptional level. And in fact, if we looked broadly across multiple cell subsets at the number of samples that significantly induced any cytokine, these were the top hits, and there's really a lot of white space. Now, it's important to note that this does not mean cytokines are not present. It means that they're not regulated at the transcriptional level by peripherally circulating leukocytes. So in fact, this is just another study we did with Angela, my critical care colleague, as well as a few postdocs in my lab and a junior faculty member in pulmonary critical care. And if we look at mild COVID in orange, severe COVID in pink, as well as other causes of ARDS or sepsis, we observe robust induction of cytokines associated with cytokine storm. So this indicates that either these are regulated at the post-transcriptional level or they're being secreted in the lung and recirculating from there. So in the last couple minutes, I want to talk a little bit about the plasmablasts and this weird neutrophil population that we observed right here. So if we go in and we just remap this on the this cluster of plasmablasts and neutrophil-like cells, we observed that they still stuck together, which we thought was a little weird. And this induced us to explore RNA velocity as an orthogonal method using the raw data to try and infer a developmental pathway and see what this would tell us. And to our surprise, when we ran this RNA velocity pathway, we observed on the inferred latent time trajectory a real time course that looked like it arose from the IgM plasmablasts into the developing neutrophils. Now, obviously, this is not proof. It is just an observation based on the RNA velocity and those limitations. But to explore this idea a little further, if we just mapped out the expression of typical plasmablast markers, as well as sequential markers of neutrophil development, it was kind of curious to us that we saw the plasmablast markers up in the plasmablasts, and then primary, secondary, and tertiary neutrophil markers being expressed along this developmental pathway. And similarly, when we did latent time analysis, we observed similar things, as well as induction of master regulators of neutrophil transcription across this latent time pathway. So at this time, it occurred to us that perhaps we were losing something because, of course, we'd like to know, do these really turn into neutrophils, and are they related to neutrophils, and can we demonstrate this bridge? But this really led to a technical issue in which it's very difficult to look at neutrophils in single cell space because they tend to degranulate if you, for instance, run them through a 10x machine. But we hypothesized that Seq-well as a well-based platform might allow us to get good quality transcription on healthy neutrophils. And so this is just a pilot experiment we did with a healthy donor in which we ran Seq-well on PBMCs. But then from the same donor, we also isolated neutrophils by negative selection. And we got actually really nice neutrophils. We also did ACK-lysed whole blood, which was fantastic because we could see all the different cell populations, including eosinophils, which we missed with that. And importantly, because it's very difficult to get high-quality single-cell reads, we observed a decent number of genes per cell, as well as unique molecular identifiers per cell in this data. So really, this has opened the door to allow us to better explore these and try to better understand this differentiation bridge. So I think I'm running low on time, and I've already gone over most of the conclusions. So I'm just going to mention that, of course, we need to better understand this, and we've got a bigger data set that we're still analyzing and unfortunately are not quite ready with, that has patients across the spectrum of disease. And we, of course, hope to better understand what the role of neutrophils is in pathogenesis. And with that, I just want to thank my amazing lab for doing this work under very short order, Calvin Kuo's lab for the organoid work, which I very briefly showed you, as well as all my outstanding clinical collaborators. And I will end there.